Welcome. Thank you for joining us. We'll give it a few moments for attendees to be able to come in and join us in this Zoom room. Thank you again for joining us for the Artificial Selection Inquiry Experiments with Yeast virtual workshop. I'll be introducing your hosts, John and Sarah, here in a moment. But I wanted to go over some housekeeping items first. We ask that you use the Q&A feature to ask your questions. Sarah and John will be answering those throughout the presentation today. And I will be including a link to a shared Google folder that includes the handouts and other files that will be used during the workshop today. So you'll have access to all of those. We are recording this session, so it will be available on our website later, and we'll post a link to where to find that on our website closer to the end of the session today. And just to let you guys know that everyone who attends live will be getting a certificate of attendance emailed to them, and we can customize that if necessary. Just email us back, and I'd like to introduce you to your presenters, John and Sarah.
Hi, I'm John Melville from Vernier Software and Technology, and I'm in the office. And this over here, or what depends on which camera you see, is Dr. Sarah Tularovic. I'm a former college professor that used to teach at Wartburg College. Sarah, why don't you go ahead and introduce yourself?
Oh, all right. I'm also a former college professor. I taught at St. Edward's University and for a very long time at the University of the Incarnate Word in San Antonio, Texas.
Great. And so what I'm going to be doing is I'm going to be taking you through an activity in our inquiry biology with inquiry book, or our investigating biology with inquiry book, activity 22, which looks at artificial selection of yeast. And I'm really glad that Sarah Tularovic is here, because she was the one that taught me actually how to brew and gave me the original idea for this lab that I wrote up back in like 2010. To just give you some background, I am a comparative physiologist. So I'm really interested in evolution, teaching evolution, things related to evolution. So what I want to do first is I want to take you through some of the resources that I've put up for you if you're interested in teaching evolution before I go to this lab. So I'll kind of walk you through the idea that I had for how to do this activity. Great. So one of the things that I wanted to let you know is you have a folder that is in chat that has almost everything that's also in this folder. I just have this folder up. Can you guys see my screen fine?
Yes, I can see it, John.
Great. What I wanted to do first was just talk about, you should see a Google Doc that has a list of resources for evolution of yeast and then resources for teaching evolution from Vernier. Now, if I hit control click to open, there's this really nice link that we have at Vernier that talks about this same lab. This is what I'm going to be taking you through, is this data, how to do this activity. This is the artificial selection of yeast lab. And then there are other resources that are all from the Investigating Biology Through Inquiry book. There's an evolution of cellulobiase, which looks at mushrooms, an introduction to molecular evolution, which looks at fish protein. That's a lab activity with Bio-Rad. And then this one here is the evolution of yeast using the CO2 gas sensor. But then there are also some other activities that we have that are not in any book. And those links can be found here. And those are exploring the evolution of temperature regulation, the diving reflex, the evolution of respiratory systems with the CO2 gas sensor that Dr. Tularovic helped develop. And then another one that looks at fish and snails as well. So there's actually a lot that you can do if you're interested in comparative physiology. Now, my original idea that I got for doing this lab activity is because in Oregon, beer is very popular. We also make a lot of wine. And I thought to myself, one of the key things that Darwin talked about when he was discussing evolution was artificial selection. So that if you look at farmers and people that also, raise different animals, they select those animals for different traits. And it can be really difficult to, actually measure or look at evolution in the lab, especially in a short period of time. And I thought, God, wouldn't it be neat if we could actually look at some of the aspects of artificial selection in a lab period? And I thought, well, maybe we could with yeast. So if you take a look at this lab activity here from Scientific American, what they did is they found a low temperature tolerant yeast that they could trace back to South America. And one of the really interesting things about lager yeast, as opposed to other yeasts, is that they can tolerate growing at slow temperatures, cold temperatures. And so that gave me this idea that maybe we could look at the temperature effect of different yeasts. And the more that I researched that, I found out that there was just a great deal of information. And so there are all of these resources that I put here for you that are in a Google Doc in that folder. There's another Scientific American article that looks at, once again, lager's yeast, then another reference that looks at lager yeast. Whoops, edit, undo that. There's even one that looks at yeast genomics and lots of other research here. What was also really interesting is they also found that in addition to lager yeast tolerating lower temperatures, there was also some evidence that beer yeast liked maltose more than things like sucrose, and that wine yeast would prefer sucrose, which makes sense. Wine yeast, they ferment on fruit. And so there's also, in this activity, there's also some evidence for that. But what I want to do now is I just want to take you through the basic activity. So I'm going to close this.
John, do you mind while you're doing that, can I launch a poll and we'll get an idea of who's in our audience today?
Yeah, go for it. I will launch GA and you go ahead and do the poll.
Okay, great. All right, I'm going to launch a quick poll here if you would just respond. I'm going to ask you a couple of questions. The first is, what level are you teaching? Is it high school, college, or something else? And then also, what type of instruction are you currently doing? Is it 100% in person, a mix, basically a hybrid, remote and in person, or are you doing 100% remote? So hopefully our participants can see that. Great, we're getting responses. All right, we'll leave it up just a few more seconds, see if we can get everybody to respond. All right, it looks like most of our audience is college. We do have high school. And then it looks like people are either 100% remote or doing a hybrid. All right, I'm going to go ahead and end the poll. Also, don't forget that if you have questions, you can certainly put them into the Q&A and I can relay them to John.
Thank you. Great. So one of the things that I want to do next is just talk about different strains of yeast. So I'm going to stop sharing my screen real quickly. Well, I'm going to keep sharing my screen, but I'm going to change the camera view. So this is a camera view here and of what's right next to me. And what you should see, this is the lamp here, but these are all different strains of yeast. And if I bring this in closer, you can see we're probably all familiar with baker's yeast right here. But this is a Nottingham yeast, a different type of brewer's yeast. Here's another beer yeast, Windsor yeast. There are both red, and this is a KIV116 Saccharomyces, which is the classic yeast. And then here's a Premier Cuvée, which would be for like making champagne. And this is all that I did when I developed this experiment, is I went out and I bought on Amazon, or I went to a local brewer and I just bought different strains of yeast. You can easily get baker's yeast, lager's yeast, different strains of yeast, and then take a look at them. Now the preparation is actually really simple. So I'm going to change the camera again, and I'm going to show you what the setup looks like. So what I have over here is I'm going to point to this. I just have some yeast right here that I've just proofed. It's about 1 package of yeast for 100 mils, and I just let that sit overnight. I have a solution of glucose right here. And then this is just a little contraption. You don't have to have anything as fancy as I have over what I have over here, but I like it. This is just a little beta chamber. So it's a little plastic chamber. I have a little aquarium heater that I can use to monitor or change the temperature of this little thing. And then I have a temperature probe here, and then I can place the CO2 gas sensor into it when I need to. Now I'm using the GoDirect sensors, which can be used wirelessly, but you can also use our other sensors as well. So what I'm going to do is I'm going to launch Graphical Analysis. And I'm going to bring it over so that you can see it. I'm just going to turn on these sensors really quick. And then I'm going to choose sensor data collection. I'm going to connect to these wirelessly. If you wanted to connect to these via USB cable, you could. I'm going to look on the back of them to make sure I get the right one. 389. And then I have a temperature probe. Now, one thing to be aware of is the CO2 gas sensor actually has a temperature sensor inside of it. So you can actually measure the temperature of the air. But in this case, I'm just going to keep on using that temperature probe. And then the instructions for this lab in our setup tell you to change it slightly. So I'm going to change this here to minutes. And then 10 samples per minute. And then for 15 minutes. And don't worry, we're not going to sit here for 15 minutes. I just want to show you the basic setup here. I'm not really interested in looking at temperature, so I'm just going to use one graph. So there's CO2. And then now I'm going to go back over to the prep, and I'm just going to show you how you do the first, what we call preliminary activity that you could show the students. Now, this basic activity that I'm going to do is just really putting some yeast with some sugar water, pouring it in the chamber and measuring respiration rate. And then you could look at different yeast strains. But it's pretty simple. Have the CO2 sensor there. Here is my little chamber. This is just a little, you could use a test tube. I like this because it's plastic and I can clean it really easily. I'm going to put 3 mils of glucose in here. Three mils of glucose. And then I'm going to put 3 mils of yeast suspension. It's important that you draw from the middle of the yeast suspension when you're doing this, because you want all of the cells to be moving around. If I was going to be doing this with students, I would probably give them 3 mils of yeast suspension that they could then invert. There we go. And then I'm just going to invert this a few times to make sure that the sugar water mixes with the yeast suspension. Then I'm going to pour it into the chamber. Now that chamber that I'm using, I actually have a little stir station on there, and I'm actually using a little stir bar. That just makes it so it responds faster. You wouldn't have to have that or do that if you didn't want to, but I really like it. Specifically, if you're teaching at a university, you probably have these floating around. There we go. I'm just going to wait a while for the CO2 level to go up. There we go, and I'm just going to hit collect. collect. Oh, hold on. Did it freeze up on me? There we go. There we go. There we go. So we're collecting data. You can see that the CO2 level is going up. I can hit this little zoom to all data button if I wanted to show that the data is actually going up. There you can see the CO2 data, the CO2 is already responding. Now, to do the analysis for this lab, you typically wait 15 minutes. Let me go back to so you can see me here. You typically wait 15 minutes because you want to make sure that the yeast have a full amount of time to respond to that sugar solution. And then you can look at things like you could look at, well, what's the response? This is at room temperature. Well, what's the response going to be at 30 degrees or 37 degrees or cool? And then you could compare different variants. So that's the basic setup that you could do. Let me stop this for a minute and let me show you something that you might like. I just did this experiment this morning, so I'm gonna open up data for this run. And one of the things that I can do, I'm using GA Pro, and if you were to get GA Pro, you could do this as well, is I am going to insert a video. So I could go to import video, and I have put a video also into that folder that you have. And it's going to take a while because this video is big. And I'm going to move it over here. And then if I tap here, this enables replay. And then I can tap here to sync data to the, let's see here. There we go, data set 1. And actually, let me disconnect these sensors here from that. Disconnect. There we go. And I'm going to say that I want to sync this data. I know that because I shot this video that the data begins at around 90 seconds. And then I'm also going to speed up the rate of play. I'm going to make it happen at 5 times as fast. So if you're teaching remote, you could actually use this video. So if I hit play now, you can see the video here. Same thing, that's me putting in the glucose suspension. Then I add 3 mils of yeast, invert, pop in the CO2 gas sensor. That little collect icon comes on. We have access to that. I can put that in there so it collects. And then there you can see the data coming across. And this is the exact same experiment that you see over here. I just did it yesterday and have the video. And so this, you're welcome to use this video. This video is in that folder. You're welcome to take it and use it to show your students how this experiment is conducted, even if you can't do it yourself. So I'm just gonna wait here. Let me know if you have any questions. I'm just gonna this is collecting data at 5 times its normal rate. That'll be a few minutes, but this would be a good time to stop if you have any questions about this process. Because the next thing I'm gonna do is I'm gonna take you through how to analyze different data sets using different yeast strains. So just let me know if you have any questions.
Joan, can you tell us a little bit about the difference between graphical analysis and Graphical Analysis Pro?
So graphical analysis does not have the ability to do video. You still can analyze everything that's here. If you guys want to go to our website, www.vernier.com, I would encourage you to download graphical analysis. I can even give you the GA Pro code so that you can play around with GA Pro for like a few days or even in this session. And GA Pro can also share data locally or on Wi-Fi, which is a really great feature. And those are the main key differences. And then it also has what we call sample experiments in it. And that was one of the things that I was going to bring up, is that there's a sample experiment that looks at yeast with a dissolved oxygen probe that is very similar. So there are also sample experiments that we've already created that you can use. So GA Pro has sample experiments. It has the ability to input video and link that to data collection. And it has the ability to share data remotely in a session, which is really useful. The other big thing is that GA Pro actually is an annual subscription, whereas just normal GA is free. Anyone can use GA anywhere at any time. But they all support data collection and putting in data, etc. Other questions?
Is there a way to try out GA Pro for free?
There is a way to try out GA Pro for free. You can actually do that now. Let me get finished collecting data here and I'll show you. If you're an instructor, if you go to the GA Pro website, you can actually ask for a free link. But what I was going to do is give you the code so to play with it for a few days, but there is a way there. So we'll go there just next. In this lab activity, what you do for the preliminary activity is you analyze this last period. So you highlight the last 5 minutes of data, and then you apply a curve fit. And so what we're going to be comparing is the respiration rate, or the rate of different yeast strains. So in this case, the slope, M, is 288, and then we can compare it at different temperatures or compare it to different yeast strains. All right, so let me stop this. And let's just take you back to the Vernier website. And if you go to products and go to software, you will see there's GA Pro and then graphical analysis. Now, if you want to right now, I would just download Vernier graphical analysis, and then I can give you the code to make it pro for like a few days. And you can play around with that right now. There's a version for Windows, Mac, Android, Chrome, etc. But if you want to get a version of this for you, of GA Pro as an instructor, all that you have to do is go down here to free trial, fill out the free trial information, and submit, and they'll probably get back to you within 24 hours. And it's a really, really nice feature. And it's incredibly affordable. This $69 fee that you're seeing here, that's for your entire department. It's not a per student fee or anything like that. It's for literally the entire department. So all students can use it, all faculty can use it in any department. Other questions, Sarah?
No, I was just going to let everybody know that Angie just put that code, that's the activation code to be able to try out Graphical Analysis Pro. This is separate than the 30-day trial. So if you want to play with it now, you can try this code first. And then if you want to play with it longer, you could still sign up for the trial.
Yeah, so what you would do is you would launch GA Pro, you would launch Graphical Analysis. And then you see this is a radio icon button. That's because I already have GA Pro. But there's a little shield that will be sitting over here. And you would tap on that shield, and then you would put in that code. And then you would get access to GA Pro. And what did I mean by sample experiments? Well, if I go to new experiment, you see here it says see experiments. There are a whole set of experiments that are here. I'll just show you aerobic respiration. Oops, there we go. So aerobic respiration has a DO probe, 4% sucrose in water, and then we have data, and then I can do the same thing. I can show that data. I'm running it 3 times. And then you could see that. So we have actual experiments that are in there so that you could give this to students and then they could analyze this data. So there are sample experiments that live in it. You can also attach video to create your own experiments. And you can also do something else called data sharing over the web, which is really useful. Here we go. So other questions before I get back to Yeast?
Not yet.
All right, so I'm gonna launch GA Pro again. And well, let me do this other thing here. Is there anyone here that uses Logger Pro? No? All right. In that folder, I have given you all of the data that I used to have for this lab, and I've got that in a Logger Pro format. And then I also have the runs themselves in GA format. So if you actually wanted to take a look at these files here, and you could use Logger Pro, you can get a 30-day trial of Logger Pro for free as well. And I can take you through those later, what those different experiments look like. But let's just take a look at some of those runs. So let's just take a look at an experiment here. I'm going to choose, what's up here? It's locking up, stop. Let me relaunch GA. Oh, hold on, I'm just gonna use the mouse. There we go. Choose file. And I'm going to choose yeast temp. There we go. So what you see here, let me make this a little bit more easy to understand. This is a series of 3 temperature runs for 3 different yeast strains. And what I'm going to do is I'm just going to turn on the graph legend, which is going to be kind of complicated. But what I have here is DLY is that's a type of lager yeast, and I looked at it at room, warm, and cold temperatures. BY stands for baker's yeast, and I looked at that at room, warm, and cold temperatures. And then this is that Nottingham brewer's yeast, and I looked at it at cold, room, and warm temperatures. So let's just take a look at one strain. So I'm going to turn off the Nottingham, and let's just take a look at baker's yeast. So if you look here, and you could open these as well. Like you could launch GA and you could open these files and go ahead and do this analysis with me if you wanted to. And I'm just going to analyze the last 15 minutes by highlighting and dragging across, and then doing a curve fit. And so I've done a curve fit for all 3 of these runs. And what you can see is, well, for the first run there, let's see, the slope really decreases. So it started off at 355 ppm per minute. And then as I look at a room temperature here, it's at 22. And then if I look at a cool temperature, oh no, if I look at it room temperature, it's 104. So it drops by quite a bit. And then at cold, it's actually at 22. Which is, to me, I think is actually kind of interesting, because that shows that baker's yeast, which you bake with, you would actually maybe expect that at a rather warm temperature, you would dramatically increase the respiration. Let's take a look at a lager's yeast. And if you remember correctly, I said lager's yeast is not very, doesn't respond very well, or it actually is not inhibited by cold temperatures. And so if you look here, this is the pink is warm, the blue is room, and that's cold. And so let's do that once again. Let's just take a look at those. I'm going to highlight the last 5 minutes, apply a curve fit. And you can see that the warm is 127, room temperature is 70, and then cool is 23. Now, any questions? One of the really big things about analyzing different yeast strains like this that can be problematic is I have no way to control, using this preparation, I have no way to control for the density of cells. Like I could measure the absorbance of those cells, but I didn't. So I don't know how many cells I have. I tried to measure out, the same amount of yeast, and then I mixed it up, but I don't know how well they grew overnight. And then you also might expect that different yeast strains are going to grow at different rates, which is not unheard of. So one of the things that we tell you to do in this lab activity is to normalize. So what you can do if you want to compare these two strains is look at the percent difference in each slope, or you would use one as a control and then look at how that slope changed. So further questions?
None currently.
So what I'm going to do is I'm going to move over to Logger Pro and just take you through some of the data that I collected or that I've observed. So let's just take a look at the effective temp. Now, once again, these files exist in this folder and you have free access to these. So this has the protocol right here. So you can copy and paste that. The data that I collected is located here. And then if you tab over to each page, there are different graphs. So this is just the percent increase at looking at baker's yeast, Nottingham yeast, and then a dry lager's yeast at different temperatures. So the effect of increasing the temperature from 14 to 33 degrees C. And you can see that the dry lager yeast doesn't really increase that much. This other brewer's yeast increases quite a bit, but baker's yeast really increases by over 1,000%. And then this is just a graph looking at those three temperatures here. Green is dry lager's yeast, red is the Nottingham brewer's yeast, and then here's the baker's yeast in black. And you can see that as temperature increases, this slope dramatically increases for both baker's yeast and dry lager and the brewer's yeast, but not for the lager's yeast, which is actually in accordance with what you would think based on the evolution of lager's yeast, which actually like cold temperatures. And then you could also look at it, they're not really, they don't inhibit quite as much when you're looking at cold temperatures. Here's another look. This is just a closer look where I actually looked at absorbance in those cases to kind of control for that. And you can see once again that even normalizing baker's yeast just increases dramatically as with increasing temp, whereas lager's yeast really isn't that much. Oh, this is the effect of salt, Logger Pro yeast. Change that there. Oh, it just went to the next. Oh, there we go. So I can take a look at what about the effect of, oh, there we go. Yeah, the effect of salt. So one of the things that you might think about was baker's yeast, you use salt when you're working with baker's yeast, and you don't typically, I don't know, Sarah, do you? You don't normally add salt to any wine yeast or anything like that, right? I mean, that's kind of
no, unless you're making a really bizarre historical recipe.
And you can see here the effect of salt on baker's yeast is it does decrease, but it really decreases dry lager yeast. If you throw salt in with a lager yeast or wine yeast, it really dramatically inhibits it. So that's another thing that students could investigate was, well, what's the effect of salt on yeast? Probably what most of you are really thinking about, though, if you've done any experiment like this, is, well, what's the effect on ethanol? Because as most of us know, brewer's yeast can't survive at very, very high alcohol concentrations.
Baker's yeast?
You could look at, well, if you compare baker's yeast to brewer's yeast and then wine yeast can tolerate much higher ethanol concentrations.
Gotcha.
And so if we look in this case, I used wine yeast and compared it to baker's yeast. And you can see that as you increase ethanol concentration using the same protocol, that the baker's yeast is dramatically inhibited. The wine yeast is inhibited, but even at 6%, it's like, no, I'm fine. And then at around, 12%, it's like, I'm still cooking, I'm good. But at 25%, you've pretty much killed most of the stuff that's there. And then this is a graph that looks at percent inhibition of different strains, and you get kind of what you would expect. Wine yeast is the most tolerant to alcohol concentrations. Lager yeast is inhibited, but not nearly as much by alcohol concentration, increasing alcohol. But baker's yeast is inhibited the most. And these are all just using that same preparation. For this experiment, what you would do is you would take, you would mix up some sugar solution in ethanol and then add the yeast to it and then measure it for 15 minutes and look at the rate. And this is all in that activity that is also in that folder. So there is the preliminary activity for students, which is a PDF. And then I have also placed in there the teacher's instructions. And the teacher's instructions from our book, Investigating Biology with Inquiry, are amazing because it actually will take you through how to do any of these activities. How to mix it up, how to prep it, ways to analyze it, how to look at percent inhibition, percent difference. So all of the resource information is there. The one thing that isn't in the current edition are the graphs, and that's why I've given you these files, is because the graphs are actually there as well. All right, let's look at one last thing. What about different sugars? So remember, and if you look at those resources that I put in there, there's some evidence that brewer's yeast and lager yeast prefer maltose to sucrose. And I found the same thing when I did this experiment, is that if you look at baker's yeast, you can see that it really prefers glucose. There's a slight decrease in fructose if you give them fructose. And this is the same molar concentration of sugars. When you go to sucrose, there's a slight decrease for baker's yeast. And, for the baker's yeast, there was an inhibition. I would probably want to repeat this over again to see if this is real. But what I thought was really interesting is that the lager yeast, there was an inhibition when I used sucrose, but it seemed to like maltose more. And then when I did the same thing comparing wine yeast to lager yeast, wine yeast is more like baker's yeast. And you can see that it prefers sucrose and fructose. And actually this fructose is actually a little bit higher than glucose, which actually makes sense if you're a wine yeast and you're used to being, fermenting over sugars from fruit. And then this is just a graph comparing all three. So you have access to all of these, all this data that you can use, play with, etc. Those are these files. I've actually also created these things that are called runs. So if you have like ethanol inhibition, if you wanted to look at ethanol inhibition or inhibition runs, that's the raw data. So I went back to data that I had and I put them all here. And then I also have in there the yeast sugar 2 is actually the video that you can actually use. All right. Any other questions so far?
Yeah, John, would you expect to see a difference between single sugars and double?
Yes, well, that's in the actual book. You really have to be careful, right? Because if you want to compare monosaccharides to disaccharides, you have to make sure that the molar concentration is equivalent, right? That's why in this case, I was working with a chemist and we basically said, hey, since if you think of a sucrose molecule, it's a glucose molecule and a fructose molecule, there's twice as much sugar. So you have to make sure that the total amount of glucose or fructose that can be utilized is controlled for. So we did that in this experiment. But yes, you would expect, like if I gave sucrose in the same percent by weight, I probably would expect there to be an increase. Even for controlling for that, you might expect there to be a decrease, because the yeast molecule, the yeast cells actually have to break down. They have to break down that disaccharide into a monosaccharide, if that makes sense. To me, this is also a great way to look at metabolism. Like, it's a super fun way to investigate metabolism. And in our Biology with Inquiry book, and in this book as well, there's a whole activity that just looks at sugar metabolism that is very similar. You can do a lot with yeast. Other questions?
Are there any sugars that you have found that yeast can't ferment?
Oh yeah, no, there are. They just didn't make it into the book because the book got really big. So you can take a look at xylose. Like right now, it's really terrific because you could go onto Amazon or go places and get all sorts of sugars that are going to taste sweet to us, that yeast will not be able to ferment. Like xylose would be one. I'm trying to think of a few other sugars that are out there that you could use. But yeah, some artificial sweeteners, or they won't be able to break them down very well. Now, what's really, really cool, or would be really cool, and I haven't had a chance to do this, is how many of you are familiar with Brettanomyces? Or have you heard of Brettanomyces? Sarah knows way more about Brettanomyces than I do, but if you like sour beer, and I do, Brettanomyces is one of the species of yeast that they use to make that beer sour. And that's because it's acidogenic. It also can break down celluloses and things like that. So it can, and it tends to live in barrels. So it can probably break down other sugars that your normal Saccharomyces cerevisiae can't break down. And you could even like compare different sugars. I know that Sarah is an amazing brewer. So when we've looked at brewing, say, like a mead or using a cider, there are things that you need to give the yeast to help them grow better, because they're not going to like growing off of that sugar source. Do you have anything else to add to that, Sarah?
Well, I was going to actually say that you brought up a really good point with talking about an acidogenic yeast, because you could also have students look at the effective pH,
yes,
on these rates of fermentation.
Yeah, that would be a great experiment, super easy to do, and you could easily use buffers. We just didn't have the time to do it. But yeah, this is a great activity because it's so simple as well. Like, all you need to do it is here. Let me stop sharing my screen because you don't need to see my screen. All that you need to do this activity is a packet of yeast and then maybe some buffers, some salt. I mean, you could do this entire activity, or a student could do this entire activity at home. You could even have them, different students, compare different things, like look at the effect of different sugars or yeast strains. I mean, all you really need is a stir station, yeast, some cups. You could easily use cups, and then hot and warm water or whatever else. It's a really, really simple and easy activity to do. Oh, and another thing that you could do is you don't have to use a CO2 gas sensor to do this activity. We have an ethanol sensor. You could use our ethanol sensor, which is actually more affordable. You could also do it in solution. So you could use a dissolved oxygen sensor. And I haven't done it. The CO2 sensor just works really well, but you could probably get away with using a gas pressure sensor and look at the amount of gas that's evolved with the CO2 that's evolved and measure that. So another really affordable way to do this would be with the gas pressure sensor. The CO2 sensor I have just found is really, really good at doing this activity. It's really reliable, it's very robust, and you get really good data. Other questions?
John, in your experience, how quickly do the cells in solution switch from cellular respiration to fermentation?
Now that's a great question. And it was really interesting to all of the brewers that I talked about, and I read a whole bunch of papers on this. What's really interesting is that I prep these overnight. So what I tell people to do is get that little stir station. Let me show you that here again really quick. I tell people to right over here, put their yeast on water, and this is in the Investigating Biology with Inquiry book. Put some yeast in here with some warm water, throw in 1 packet of sugar, and let it sit overnight. And that way you've made sure that everything has gone over to fermentation, if that makes sense. But what I have found in the research that I've done and looked at is that, yeast, Saccharomyces cerevisiae in particular, is going to be mixed, but it's going to prefer fermentation all the time, even if there is oxygen around. And that's because it's wanting to grow rapidly and fast. So even in cases where you would think there was some oxygen available, if there is an excess of sugar available, they are going to ferment, or they're going to primarily ferment. They're not obligate, right? They're not one or the other. They can do both simultaneously, and or they can switch. But I make sure that they're all fermentative by letting them sit overnight. And that's actually a great thing to do with the dissolved oxygen probe, like I did in that activity that's in GA Pro, because you can see that if you just put a little bit of yeast in some water, the amount of oxygen in that water disappears really quickly, on the order of minutes. So even though there might be oxygen in the atmosphere, there's not any oxygen in the water that the yeast are in. Other questions?
Doesn't look like we have any more audience questions. Did you think of any other options that students can test? That was quite a few parameters there.
Oh, I mean, what I would do is take a look at the Investigating Biology with Inquiry book. In that folder here, let me show you that to you really quick so you see this resource. Let me share my screen again, because I don't think that people understand what an amazing resource our books are. And then I also can show you how to get access to your resources as well. So let me share my screen. I'm going to open up. So this is the instructor's guide. And in there, it has an overview. I have we even talk a little bit about Darwin's theory of natural selection. We provide some resources. We provide the learning outcomes. We talk about the inquiry process. We give you all the materials. We talk about the preliminary activity, show you what you should expect, how to perform or calculate the percent difference. But then what we really do, which is really important in this activity, is we go through all of the researchable questions that students can ask. And they start off from simple to more complex. So if you're teaching a basic introductory biology class or an advanced one, you can make them more complex. So as you see in here, we've got how do respiration rates of baker's yeast and lager yeast and sugars compare, just looking at basic temperature. For advanced students, we have ethanol and salt tolerance. And then looking at sort of an advanced version of that. And then once again, as Sarah brought up, we do have looking at pH. But there's lots and lots of different things that you could look at. And then we also give you the data and graphs. Now this is from the first edition. So I've given you this instructor's edition because it actually has the graphs in there. The newest one doesn't have the graphs, but you can have some examples. And then what's also great is we tell you how we actually did this lab experiment so that you can make it repeatable. Now, I don't know if any of you have any of our books, but one of the great things that you can do is if you have purchased one of our books,
or if you think somebody on your campus has purchased one of our books.
Oh, if you think of someone, you can log into your account, or you can create an account. And this is a little bit, did it get my, yeah, this is a little bit tricky because it'll look like nothing changed, right? Like I logged in and nothing happened, except now it says my account. And then there's my saved carts, account details, but most importantly, my downloads. And if I go to my downloads, it will show you the software installers that you have access to, the subscriptions that you have access to, and then also the lab book electronic resources, which are really important. And you can see I should have investigating biology here. There it is. And you can download the entire electronic resources right there. So anytime that you need them, you can get them. So this is a really great feature that we have on our website. Other questions?
So how did book licenses work for high schools as opposed to college, John?
I believe it's for book licenses for high school are for building. And then for colleges, it's per department. And the site licenses work the same way as well. So if you got Logger Pro or GA Pro, that's for the department. Whereas GA Pro for like a high school would be for the entire building. There are district-wide licensing, things like that. So if you're like a large community college or a series of colleges, or you wanted to roll it out larger, just get in touch and we can talk about that.
And can faculty freely copy the student instructions and distribute them to students?
Oh yeah, yeah, no, there is no copyright issue. Once you've got the book, we assume that you want to be able to use it, push it out, etc. In some cases, when I was teaching at Wartburg College, and I know that Sarah Talarovic did this when she was at Incarnate Word, is you could even take our instructions, put them in your lab manual, and you can give that out to students as well. And it's fine if students have to pay for that resource. We just don't want you republishing our lab manuals and charging people for it. We don't want you going to Pearson or anything else, but you're welcome to give it to students for free. You're also welcome to put it into your lab manual, even if students have to pay for it. Well, that's all I had. What I wanted to do just once again, though, was just remind you that Darwin Day will be coming up in February. And you can always take a look at these evolution resources. Where did they get? Right here. You have them in a Google Doc. This is, but they're all listed right here. And oh, one other really cool thing I wanted to give Dr. Talarovic some big kudos here is her activity is also linked here. So if you just want to introduce your students to evolution, there's this great article by Sarah Talarovic on introducing evolution with candy and then deepening student understanding. All right, so this, I'll go back here again. So this is located in that Google Doc that you have. And if you go to other activities and click on it, it will take you to this great blog post on how to introduce your students to evolution that Dr. Sarah Talarovic wrote. And it's a really great activity looking at candy. And I'm sure that you could also do this remotely, and I'm sure that students would have no problem playing with M&Ms remotely as well.
Yeah, and that's actually not I am not the first person to do the evolution with M&Ms. But we shared it as a great icebreaker for the classroom. I used to do that every semester.
And then last, let me show you some the other activities here. Control. Other really simple activities that you could do. These are all in, these activities listed here are all in investigating biology through inquiry. But these other ones are actually really simple. Like you could look at the evolution of temperature regulation in mammals. That just uses a temperature probe. And you just mimic blubber and fur. It's really affordable. This is a neat one if you're a biologist and you like cockroaches and scorpions. This is a lab that Dr. Talarovic did. looking at respiration rate in scorpions, which have a really, really, really low respiration rate relative to cockroaches. And then this one is actually really simple too, and would be really simple and straightforward, is you just take a dissolved oxygen probe and you look at how much oxygen is consumed by a snail, by a goldfish, and then by a fighting fish or a betta. And then you can talk about open and closed circulatory systems and how they evolved, and then behavioral adaptations and evolution, which are also pretty cool. And that's pretty much all we have for you. Unless you have further questions, feel free to get in touch with us at any time. You can always email us at biology@vernier.com, and someone in the biology team, you have been talking to 2/3 of the entire biology department. Well, Robin Gastineau is also a member of our biology department, so we can actually consider her maybe another third, but there you go. So if you have any other questions, please let me know. But if you ever have any questions about biology, you can email biology@vernier.com, and someone from the biology team will get back to you. And thanks.
All right. Thank you so much for attending today and our broadcast.